Nobel laureate Professor Horvitz, Yang Berbahagia Datuk Engineer Dr. Haji Ahmad Zaidi Laidin, Vice President, Academy of Sciences Malaysia, Mr. Uwe Morawetz, Chairman, the International Peace Foundation, Academician Professor Dr. Lui Lai Meng, Senior Fellow and Council Member, Academy of Sciences Malaysia, academics and researchers from institutions of higher learning and research institutes, officials from government ministries and agencies, distinguished guests, ladies and gentlemen. Good afternoon and welcome to this dialogue with Nobel laureate Professor Robert Horvitz on regulation of organ development and program cell death, organized as part of the Malaysian Bridges event series facilitated by the International Peace Foundation and organized by the Academy of Sciences Malaysia in collaboration with University of Malaya. Ladies and gentlemen, without further delay, it is now a pleasure to call upon Yang Berbahagia Datuk Engineer Dr. Haji Ahmad Zaidi Laidin, Vice President, Academy of Sciences Malaysia, to deliver his speech.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon. Professor Robert Horvitz, Nobel Laureate in Medicine for 2002. Mr. Uwe Morawetz, Chairman of International Peace Foundation. Academician Professor Dr. Lui Lai Meng, Fellow of the Academy of Science, Council Member, as well as the moderator for this dialogue. Fellows of the Academy of Sciences, Malaysia, Yang Bahagia Tan Sri, Dato', distinguished guests, ladies and gentlemen. Well, first I'd like to greet you all a very good afternoon and welcome to this dialogue. It is indeed the third in a series of dialogues with the Nobel laureates organized by the Academy of Sciences as part of the Malaysian Bridges program of dialogues towards a culture of peace. I would also like to say a special word of thanks to the University of Malaya, for playing host on this historic occasion. But I do know that the top brass of the university are otherwise engaged with the Honorable Minister of Science today. So that's why you do not see the Vice Chancellor or the Deputy Vice Chancellor here. Nevertheless, we are indeed thankful for their hospitality. The Malaysian Bridges series of events is a remarkable series, as it provides a platform to hear from an impressive array of speakers, ranging from Nobel laureates in physics, chemistry, medicine, economics, and peace, as well as world-renowned artists and several prominent international personalities. And so far, over 20 Nobel laureates from various fields of sciences have come to Malaysia to deliver lectures and to contribute their expertise under this program, the aim of which is to encourage the achievement of excellence in science, technology and innovation, and the development of high-caliber scientists. Ladies and gentlemen, I was privileged this morning to listen to the Nobel laureate, but I was even more intrigued when I read his short autobiography, his autobiography. And among other things, I found out that he was managing editor of the MIT student newspaper. And of course he was a brilliant student, you can tell from what you read here. But there is this particular paragraph that I thought very intriguing. It says that my coursework probably suffered because he is involved in so many of the students' activities, but not my grades, particularly what I was learning And I will also wait until the night before an examination, stay up at night and learn all that I need to know to answer the questions the next day. And to me, there has been this dream of every student who want to be able to do last-minute work and still get good grades. But obviously, not so many can do it. So our guest this afternoon is one of those. He actually majored in mathematics and economics for his first degree. And therefore, it's very unusual. I think, sir, you're one of the few, if not the first, that I've met where you started with economics, but ended up biology. And more than that, getting a Nobel Prize in medicine. He will tell you later that he's not even a medical doctor, but that's another story. What I found very intriguing is he's a man of many parts. He's also been you've worked in an insurance company, if this is true, I'm sure it is. And also an IBM, he worked for a while in IBM as a computer scientist. So, actually we have a remarkable person here, almost a Renaissance man in the 21st century to be addressing. So, ladies and gentlemen, I was privileged this morning to listen to what he had to say. And believe you me, he was extremely focused. He didn't touch anything at all about his poker playing days. He didn't talk about his bridge and other things. But all he did was to focus on science and on the research that he did. And he's got a lot to say. It is indeed a great pleasure for us to be here. And I think it is also our good fortune, that your good fortune, that so many other students that I see, as well as people from other fields to be here today. Ladies and gentlemen, I don't want to take any more of your time. I would just like to convey the Academy's sincere appreciation to the International Peace Foundation for facilitating this series of events, as well as to everyone who had contributed in one way or another to make the series possible. And so, ladies and gentlemen, I would still like to repeat our grateful thanks to the University of Malaya for their collaboration and partnership. So thank you very much, and we will look forward to hear from you, sir.
Thank you, Dr. Zaidi. Ladies and gentlemen, we now invite Mr. Uwe Morawetz, Chairman of the International Peace Foundation, to deliver the speech.
Increasing ethnic, political, religious, and social conflicts, and the helplessness to respond to them without further stimulating the spiral of violence show us daily peace is not really here. It has to be constantly learned, experienced, and created anew. Peace is also not a passive state. Peace is a process which needs time, attention, and the participation of all of us. And peace is with education. The seeds of peace need to be planted in schools, in universities, in the new generation. This is why the International Peace Foundation cooperates with major schools and universities, as well as with UNESCO in realizing this bridges program. We believe in the importance of the education and scientific community of Malaysia by bringing the knowledge and wisdom of Nobel laureates for peace, physics, chemistry, medicine, and economics to this country. What these highly in-demand Nobel laureates normally see of a country are airports and hotels. They deliver the lecture, stay for one night, and leave. We have invited them here not only to speak, but to share, to engage, to listen. They come here not for one day only or one event, but for various dialogues in different parts of the country, without requesting any honorarium because of their real interest in building bridges, because of their real interest in Malaysia. It is our hope that these are not the first and final visits of these Nobel laureates, but visits with many fruitful returns to build long-lasting friendship and to start, for instance, research programs or other forms of cooperation with the local universities and schools. This is how bridges could develop into a long long-term and sustainable success for Malaysia to strengthen education as a basis for peace. I thank the Academy of Sciences of Malaysia and the organizing committee of today's event for taking up this opportunity of our cooperation and for inviting today the 2002 Nobel Laureate for Medicine, Professor Horvitz, to this prestigious institution. We look forward to this dialogue, to his important contribution to great relations. Welcome, Professor Horvitz.
Thank you, Mr. Morawetz. Ladies and gentlemen, it is now a pleasure to call upon Academician Professor Dr. Lui Lai Meng, Senior Fellow and Council Member of the Academy of Sciences Malaysia, to introduce the Nobel Laureate and moderate the dialogue.
I'd like to add my welcome to all of you to this afternoon's dialogue. It is not easy to give a brief introduction of somebody as distinguished as Professor Horvitz, but I will try to be brief because the purpose of the dialogue is the dialogue to have a chance to probe and to discuss issues with Professor Horvitz and not to listen to me. Nevertheless, I think it is important to know a bit about Professor Horvitz, and I hope you'll spend these few minutes to say a few words about him. Howard Robert Horvitz was born in Chicago, USA in 1947. In his autobiography, Professor Horvitz paid very warm tribute to his parents, describing his father by profession and his friend. As a humanitarian, he cared deeply about people, and his mother, a science teacher, as a wonderful model for the professional woman who was dedicated to both family and work. His parents created what he described as an environment filled with love of knowledge and a joy of inquiry and discovery about the world around us. In this environment, young Bob Horvitz went through the usual skirmishes of boyhood, stitches included, but clearly he had a curiosity and questioning mind that went beyond the ordinary boy. In his 9th grade, he used his mother's bedroom for Drosophila breeding experiments to replicate Mendel's famed inheritance ratios. And it is indeed a tribute to his mother that such a messy and even smelly experiment should be allowed in the family bathroom. I'm not sure how many mothers will allow that today. The project won a prize and also earned him a trip to the Illinois State Science Fair. And from then on, I believe, he was hooked on biology. And so began the journey that would lead him to the Nobel Prize in Physiology or Medicine in 2002. H. Robert Horvitz received his PhD in 1974 from the Harvard University, and he joined the faculty of the MIT Department of Biology in 1978. He was named David Koch Professor of Biology in 2000. He's also an investigator at the Harvard Hughes Medical Institute and in the Government Institute for Brain Research. Professor Horvitz's research is focused on the genetic programs that control cell death. To unravel this, he has used a variety of techniques that we should learn from this: genetics, molecular biology, biochemistry, electrophysiology, laser microsurgery, pharmacology. There should really be no boundaries as to what techniques we use when we investigate something. He centered his studies on the tiny, almost microscopic and transparent soil work that nematode sea evidence. In 1986, he reported the discovery of the first two death genes, SET3 and SET4, which participate in the cell killing process. And later, he showed that another gene, SET9, protects the killed cell. He has also shown that most of the genes involved the lowly soil worm have counterparts in humans, and those of us in medicine would recall the Banks genes and the CL genes. We appreciate today that programmed cell death or apoptosis is fundamental to developmental biology and anatomy. That during fetal development, large numbers of cells and tissues must die so that functional structure and form can emerge. Somewhat like, perhaps, the creation of a beautiful sculpture through careful chiseling away of stone. So then, misregulation of programmed cell death could lead to inappropriate extension of lifespan of cells, giving rise to abnormal structure and function, and increasing the risk of mutations, cancers, autoimmune diseases, neurodegenerative diseases, to speak of a few. Indeed, Professor Horvitz's work has opened a whole new field of the pro-apoptotic and anti-apoptotic control of cell death with numerous potential exciting applications in science and in medicine. And this rich, exciting field is the topic of this afternoon's dialogue. Professor Horvitz.
Thank you. Thank you to all who have spoken before for their kind words, the introduction to me, and the introduction to this afternoon. The operative word this afternoon, as I understand the goals of the program, is dialogue. So I do not intend to sit here and lecture. I gave a lecture on the campus earlier this morning, but rather to say a few introductory remarks and then hope very much that we will spend the afternoon in dialogue because my goal is to learn from you at least as much as I'm capable of conveying. As Uwe Morawetz said, oftentimes, all too often, one is invited as a speaker, one flies in, speaks, and flies out, and besides frequent flyer miles, very little is accumulated by a speaker invited in that context. So what has been special to me so far in the 2 days that I've been here is the opportunity to discuss. And so I'm very much hoping with the people who are here that we will be having a dialogue. Now, having said that, the title of this afternoon's discussion is Regulation of Organ Development and Programmed Cell Death. This phrase comes directly from the Nobel citation from the award of the Nobel Prize in Physiology or Medicine that was given to me and to two of my friends and colleagues, John Sulston and Sidney Brenner, in Stockholm in 2002. And so I would like to say a little bit about the science related to these topics and try to put that into the broader context of science and science policy today. So as you have heard, the primary research for which I was honored was research, despite the fact that the Nobel Prize is in medicine, was research that was carried out using as an experimental organism a microscopic roundworm. Now, many physicians know about roundworms or nematodes because these organisms are very widespread, and they are key as parasites. They are key in plant parasitology. They are key in animal veterinary parasitology. And of course, physicians know them best because many of them are human parasites. The nematode, the roundworm that I study, is free living. Okay, now that phrase doesn't mean that it lives in San Francisco in the late 1960s, but rather means that it is not a parasite. It lives in the soil, and in fact, if we wanted to, we could go outdoors here and probably collect C. elegans from the soil right outside this building. It is microscopic. It is 1 millimeter in length, so that if I were to show it to you and you were to hold a Petri plate with this nematode on it, you would see these organisms looking to be about the size of a piece of lint. But if you look carefully, you will see that those pieces of lint are moving. So it's a microscopic organism, and it's a worm. And the question is, how did one go from research on a microscopic worm to the podium with the King of Sweden in Stockholm in the year 2002? And the short answer to that was by doing basic research involving two different biological problems. The first problem was that of organ development. And the studies that my laboratory, and then after that the laboratories of in particular one of my students, the studies that we carried out were of the development of a sexual organ of this nematode. Now the interest wasn't in the organ per se, but rather in the general problem of how do organs develop in any creature, be it a tiny nematode or something more complicated such as ourselves. And in analyzing this problem, what we found, and now by we I mean collectively the field, because the work was not entirely done by my laboratory, what we found was, first of all, in development, cells talk to each other. Cells, of course, are the unit of life, and organisms are composed of many cells and a diversity of cell types. And as organisms develop, these cells are not autonomous, but rather they interact. And some can signal to other cells, some can receive signals, they work together, they can form structures together. And it turned out in the development of this organ, there was a particular signaling pathway that was of pivotal importance. We analyzed the signaling pathway. We identified genes that are responsible, and one of the genes that we found turned out to be familiar to a human gene that had been identified very shortly before. That human gene is known as RAS, and RAS was the first human cancer gene that had been identified. So suddenly, by studying the development of this worm organ, we were studying a set of genes that included the human cancer gene. One of the biggest problems in RAS cancer biology of the day was how this RAS gene functions and what controls it and what does it control. And the answer to that came from a combination of experiments done in part from studies of the mammalian RAS gene, of mammalian cells, mouse and human, that were grown in Petri plates, from studies of another aspect of animal development, namely the development of the eye of the fruit fly Drosophila, experiments primarily carried out at the University of California, Berkeley in the United States. And thirdly, from studies that we and one of my ex-graduate students did studying this nematode C. elegans. The remarkable conclusion of these studies was that the pathways that were emerging from studies of human cells, fruit flies, and roundworms pathways turned out to have the same genetic components. And in fact, as a discovery was made using one of these experimental systems, it could be immediately translated to the others, thereby facilitating studies of the other system. And in a very short period of time, we went gene to gene, organism to organism, and worked out the pathway by which RAS functions. So suddenly we had a pathway for a cancer gene, and this pathway has turned out to be pivotal in the development of a variety of relatively new anti-cancer agents. And from our efforts, the results came from studies of organ development. Now, in parallel to this work in my laboratory, we had a number of other projects. One of these projects focused on the problem of programmed cell death. This is a phrase that is often spoken synonymously with the word apoptosis, and I can say that that word is pronounced in at least 7 different ways. So for the scientists and clinicians in the audience, don't worry if you say it some other way, because someone else no doubt says it just like you do. So that is approved. My colleague John Sulston had discovered that during the course of the development of the nematode C. elegans, more cells are generated than are found in the adult animal. And those extra cells, if you will, die. And this death then is a normal part of the developmental program of the organism. And we decided to ask the question, how do those deaths come to be? What is the control of those deaths? So what we did was to seek, identify, and characterize genes responsible for programmed cell death. And as you heard a few moments ago, we were very fortunate in our study. We identified a pathway of genes that are responsible for this process. It turned out, something we didn't know ahead of time would necessarily be the case, it turned out that these genes are conserved amongst animals, including ourselves, so that the pathway of genes that we found to regulate programmed cell death, to regulate apoptosis, and the nematode C. elegans corresponded precisely to a pathway of genes in us. Now, people are more complicated than worms, and the genetic control of programmed cell death or apoptosis in humans is more complicated. In many cases where there's one gene in the worm, there's a family of genes in people. But the genes are similar to the worm genes, and the ways in which they act and interact with other genes are very comparable. So the pathway we discovered turned out to be a pathway that was conserved in people. This pathway is important in human biology. When this pathway is disrupted, it turns out to be responsible for disruptions for a variety of human diseases. In fact, many different disorders are disorders in which there is either too much or too little programmed cell death. Too much means that this program for cell self-destruction, suicide, is unleashed in the wrong cells at the wrong time, causing them to die. Too little means when cells should die, they don't, so that cells that should be removed from the body instead survive. For example, in our immune systems, we are, as we sit here, there are cells within our immune systems that are undergoing programmed cell death. And the reason for many of these cells is that if they were left in our bodies, they would recognize our own bodies and attack them. We have mechanisms to get rid of those cells, and those mechanisms trigger the process of programmed cell death. If that process is not triggered, those cells survive. The consequence, autoimmune disease. There are similar examples in cancer where cells that should be dying instead survive, and that promotes the cancerous growth that typifies tumorigenesis in the vast majority of human cancers. So what we learned was relevant to human biology, was central to a broad variety of human diseases, and again, as we heard, has paved the way to a set of programs in the pharmaceutical and biotechnology industries focused on the goal of intervening with the gene products, the proteins of these genes that are responsible for programmed cell death. The goal being, if you can block cell death in diseases in which there is too much, or if you can activate cell death in diseases in which there is too little, you have an opportunity to have a therapeutic benefit. So that is a summary of the science of these two areas. Of course, there is much more detail. One could go through data and micrographs and graphs and tables and numbers and statistical errors. But the bottom line is we studied a tiny worm, we studied basic science, we discovered pathways and processes that are universal amongst animals and that are central to very important human diseases, and our findings are being used as we speak to develop novel approaches to therapy. The message of all of this that I think is most simple is that the most basic of research can have impact in the most practical of ways. And so that one wants to recognize that basic inquiry is important both as an aspect of human endeavor. The pursuit of knowledge is a very uniquely human endeavor and extremely important. And that pursuit of knowledge, of course, leads to discoveries and the potential for innovation that in turn can help us as a human species develop in a practical way applications that will improve the quality of life of people around the world. In the context of the International Peace Foundation, my belief is that trying to work to improve quality of life, to improve communication, to improve health, to improve education, all of these are goals that are essential in terms of trying to reach this goal of world peace. So I think I will leave with these very basic introductory remarks and hope now to turn to the dialogue, where I would be very delighted to hear comments, perspectives, respond to questions in any way that might be helpful.
Thank you, Professor Horvitz, for that very succinct account of program cell death. I'd like to open the dialogue now. We're happy to take questions. There are microphones at the desk, as well as microphones, standing microphones for anyone at the back who would like to ask any questions. Please identify yourself very briefly and make your questions brief as possible, so that we could run to a lot more discussion. Any starters? Yes, okay, please.
Thank you so much. Good afternoon. My name is Azizan. I'm from the University of Malaya. I work at the Center for Civilizational Dialogue, and my background was biology. So this morning's lecture was a very, very enlightening and it was a very happy moment for me to hear our professor to talk about science and peace and prosperity for the whole of humankind. So my point is, there seems to be, in the minds of many people, a huge gap between the natural sciences and the humanities and social sciences. So in one respect, this is an issue because Professor Horvitz also talked about how science can influence policy. And I think half the time, many people in the political area, they may not be familiar in the sense of being able to appreciate what the content of science is. And I think that's a real situation. And there's also talk that natural sciences is becoming not so popular among the younger generation. So my question is, Professor, can you sort of provide us some insights, or just from your own experience, how do we, first of all, overcome that gap? How do we narrow that gap? And secondly, how can we make people, the lay people, society at large, to have greater appreciation of the contents of science? Because science is very important in the sense of, let's say, climate change at the moment. There's enough science, but there's still not enough efforts to change habits, for example, towards the improvement of climate change. Thank you, Prof.
Okay, so there are two different, but actually I think very much related questions. One is, how can scientists and science interface with the policymakers of government? And two is, how can science and scientists help educate, inform, and interest young people? So let me turn to the first question first, again speaking simply from experience. So both of these questions are, needless to say, not unique to Malaysia. They are questions that are major around the world, and in fact in the United States, it's been very clear that both of these are substantial issues. One approach that I've been involved with personally for 20 years now is based upon a group of biomedical scientists who decided that the so-called ivory tower of academia was not an appropriate base for communication. The group of people involved initially were 4 scientists from the University of California, San Francisco. Harold Varmus, Mike Bishop, Bruce Alberts, and Mark Kirshner. Now, I don't know how well these individuals are known here, but all of them have since then gone on to do other things, sometimes with very major roles. Bruce Alberts became the president of the United States National Academy of Sciences, and Harold Varmus became the director of the U.S. National Institutes of Health, for example. And what they decided was that what was necessary was to establish a vehicle for communication with the United States Congress. And they established an organization which was initially called the Joint Steering Committee for Public Policy, and is now called the Coalition for Life Sciences. And this organization has as a major goal education of members of Congress and advocacy for support for biomedical science, and more generally for disciplines that interrelate with biomedical science, which today I would say is essentially all areas of science and many areas of engineering. The group used as its basis some of the scientific societies: American Society of Cell Biology, the Genetic Society of America. I was president of this society for some time, so that was part of the reason for my involvement. And other societies, Society for Neuroscience, and I can go on and list them. So we had as a base a very large number of scientists within the U.S. And then we went to Washington, and we did a variety of different things. One is we met with members of the Senate and House of Representatives, namely with our representatives in the U.S. Congress, and we talked to them about science and science policy. We helped Congress establish a so-called congressional caucus, and what that is a meeting that's held about 8 times a year, where a member of the scientific community comes and speaks and talks about some advance in science and how that advance in science might be relevant to thinking about aspects of policy and aspects of funding. For example, I gave a congressional caucus in which I explained about genes, and I explained how the genes in very simple organisms like a yeast cell or a fruit fly or a roundworm were actually exceedingly similar to the genes in us, and how we could study genes in these simple experimental organisms and understand human biology and human disease much more efficiently and much more rapidly than we could by trying to study the disease process itself. And furthermore, how the discovery process might lead us to an understanding of disease when at first we wouldn't even have known that the biology was relevant to the disease. So there are caucuses of this sort, about 8 per year. These caucuses have evolved into the most popular congressional caucuses. We have a greater audience and a greater participation. The topics are highly varied. Needless to say that members of Congress are human beings, and they are interested in science to varying degrees, and in disease in very specific ways. So there are diseases that are, of course, of great interest to different members, and there's some areas of science, like aging research, that seem to be of interest to all members of Congress. So we are careful to choose topics that we think will be interesting and to choose speakers we think will be interesting. And the speakers come and give presentations and then also meet individually with members of Congress chosen appropriately. In addition, in the U.S., there are oftentimes laws that are coming up or policy decisions that are being made that impact on science. And so this organization keeps an ear carefully tuned to what is happening in terms of laws and policy decisions. And when there are decisions that are being discussed, we make sure first that we have experts available to come to meet with people, to testify before Congress. And secondly, we have a system that is now email-driven, used to be fax-driven, and before that was more complicated. That's a rapid response system, so that if there's something coming up that we think is not appropriate, given what we know about the science and what we think would be appropriate science policy, we can get many, many scientists from across the country to contact the relevant people, the people who are closest to them in terms of being representatives, the people who are closest in terms of fields of expertise. And there are other things that this organization has done. You can read about all of it on the web under the title of the Coalition for the Life Sciences. So this organization is one that was started by basic scientists and has been used by the scientific community to directly influence science and science policy in the government. Okay, now, the question about influencing young people is also one that is extremely important. And I think, from my own experience, one of the most important things that's been ongoing in the United States is the support of young people who have interest in science through projects and science fairs, and essentially raising money from industry to support science fairs and science projects for young, in this case, high school students. So just a week ago, there was the Intel Science Project Fair. It used to be a Westinghouse Science Project Fair. And young people from all over the United States come to this, and they get to meet scientists, they get to present their science, and they derive from a much broader base of young scientists who have presented science projects, much as I did with my Drosophila project, although I didn't win when I was younger. So I think that's important. I've been involved in that, and I've worked to help foster that program, but it is not sufficient. One has to start younger, and one has to act more broadly. There has to be really a culture of science that is developed at the national level. And that means promoted by national leaders, promoted by science leaders within a country in ways that reflect an excitement of and a respect for science. Now, that can be a very difficult thing to achieve. And I would say that we're struggling right now in the United States to find a way to do that again. It has been done. Some years ago, during the era of President John Kennedy, there was a very conscious effort to stimulate science, and it was done around the space program. And many young people got excited about space exploration, and they ended up as scientists, not necessarily studying space, but because when they were 7 years old, they were 10 years old, they got excited about space, and then began to think about problems in science. We need equivalent things that will interest our young people. And of course, we have to couple that with schooling that stimulates creativity rather than stifles it. So the way to learn science is not, the way to learn what science is really like is not to take a textbook and memorize long lists of words and phrases, but rather to do an experiment and to observe something with your eyes based upon what your fingers have done, and engage the young people actively so that they're not sitting and being told, but they're rather doing and discovering. And to develop educational programs that are very active programs where the students can make discoveries and identify with them as those discoveries being their own. Again, there are a variety of ways of doing that, and I think the simplest statement is it's not simple. There isn't a short answer to how to really try to turn around a culture that is growing to be a-science or even anti-science, but to think hard, particularly about the youngest people, what would make them interested, what would make them excited. Bring them to your laboratories, show them what goes on, make it real, and that can go a long way.
First and foremost, I'm Vincent, Dr. Vincent from Malaysian Senior Scientist Association. First and foremost, I'd like to congratulate Professor for an excellent discovery. I think we will miss the boat now if we don't ask this question, because your discovery is remarkable. This discovery, I think, is a eureka. Bravo means which you have a breakthrough in science, that much of the scientific books have to be written. Especially, this will be determine the cure for cancer, and which is coming to be the #1 cause, killer cause for mankind. So now the question is, there are two questions here. I'd like to ask is, how would you, when will this program of cell death and gene, cancer genes, all this can be used to be utilized in the future? Or is it available now to be used for treatment of cancers or to overcome problems of humanity And secondly, I believe there will be more than one genes. There will be more than one genes that will be for prevention of cancers. Because I think it could be a multiple cause of genes rather than one gene which you have discovered or two genes. And of course, lastly is how would your cells stand up with the challenge of the causes of various causes of cancer? Because there are many causes of cancer. I mean, there are varying causes of cancer. So your programs, your cells programs, and all cancer cells or whatever, how can we stand all these challenges? It's not only just free radical that cause cancer. There are many other things that cause cancer. So how we match up your genetic things with the various causes? Thank you, Professor.
Okay, so I think there were three separate questions there, and I will respond to all three first in the same way. And that cancer is not a single disease. There are many different cancers, and these cancers can be as different each from the other as any given cancer is to a variety of other diseases. So as we think about developing novel therapeutics, we can't lump cancers all into a single word or a single entity. We must understand the different cancers. And we can see that from the biology that we know today. We can see that from the therapeutic perspective. There are treatments for some cancers, and there are no treatments for other cancers. So we know both by the biology and from the clinical medicine that we are dealing with very different entities. In fact, even those cancers that seem to have key words that put them into a single entity, like breast cancer, we know as molecular studies are proceeding, even these kinds of cancers can be subdivided into more groups. So we have to remember not to think about cancer as one disease. Now, the therapeutics that are being developed, there are many therapeutics that have been developed and many that are underdeveloped, that are under development, and many that are envisioned in the context of the different cancers. So, for example, you have a treatment like Gleevec, which is a treatment for one cancer, particularly it was developed for chronic myelogenous leukemia, CML, and it works extremely well against that disease. And the development of this treatment was totally based on the scientific understanding of the protein that drives the disease. So the science can lead to the identification of a key gene and a key gene product, the protein, and then one can do the chemistry and the medicinal chemistry to develop an inhibitor and get a compound that can be a good therapeutic, as Gleevec is for CML. Okay, now having said that, we can go in two directions. One is that Gleevec doesn't cure all cancers, but secondly, it's actually being used to treat more than the original cancer that it was developed for. So there is an increasing breadth of use. Many other therapeutics have been derived to treat cancer. So for example, in the context of the two stories that I alluded to, worm organ development and the pathway of the RAS cancer gene oncogene, there are compounds that are used to intervene with that pathway that are acting against an early step in that pathway and are useful to reduce some of the effects of that cancer pathway, but they are by no means cures. And there's very active research now to find better ways to intervene with that pathway. The pathway of apoptosis, of programmed cell death that I alluded to, there are no therapeutics based upon that pathway that are clinically proved and clinically used as marketable products, but they are very much in the testing stages in clinical trials. So they are underway. And which cancers cancers they will be most useful for is something that is still being ascertained by the pharmaceutical industry. So I think the short answer to your question is we have to separate the cancers into their various types, understand as much as we can about their biology, try to develop compounds that will intervene with the specific biologies, test them appropriately, and for those that seem efficacious, test them more broadly to see if they could be of utility in other cancers for which perhaps we don't know as much to decide what route to take.
I'm from Institute of Medical Research, Kuala Lumpur. Thank you, Prof, for highlighting the coalition of life sciences, and hope that one day the Academy of Medicine, Academy of Science in Malaysia can take up the challenge to have a similar program to directly address the government in Malaysia to advocate scientific development. I have three questions. The first is, so that visits by top scientists will not be just a one-off event. It will be nice to set up an international research corridor, so where international visiting top scientists can set up their laboratories alongside with the local researchers in Malaysia. I'd like to know what would you say are the most three important aspects as prerequisites for top scientists to want to set up a lab here? That's the first question.
I think that would vary among scientists, but basically the opportunity to pursue a scientific dream would be very important. So that means the appropriate support, and the support would include financial support, but not be limited to financial support. One would want to have an infrastructure in place where there are resources and colleagues who would be stimulatory and synergistic with an individual. The best exchange programs and visiting programs are those in which one doesn't simply go from one place to another and do exactly in the second place what you could have done in the first place, except with more difficulty because you're no longer at home, but instead to come someplace new where there are new opportunities for interactions and new opportunities for synergisms and new opportunities to develop programs that one couldn't do at home. As a model for this, I would say you could look as close as Singapore at some of the programs that have been developed in Singapore, where there have been scientists from outside Singapore who have come to Singapore to set up laboratories, to set up collaborations, sometimes full-time, sometimes for 6 months of the year, sometimes for 3 months of the year, but always with an eye to being able to do more and different than what they can do at home. There has to be something that is going to make for a greater opportunity in terms of the science.
And so will people like you be open to the possibility of being a member of a scientific advisory board of medical institutions?
I am a member of a very large number of scientific advisory boards. So I would say the answer is yes. I've certainly accepted many such responsibilities. I enjoy doing that, and I like, if I feel I can provide helpful input to doing so. I always have to decide, because time is limited, whether a particular opportunity can use my knowledge in the best way. But in principle, I try to do that. I've served, for example, many years on advisory boards in China to help with some of the programs there.
Actually, my second question is, can you comment on the use of C. elegans as model organism for high throughput screening for drug discovery of targeted therapies?
So for high throughput screening, and that word means, that phrase means different things to different people. So if one talks to academicians, usually it's quite different what they have in mind than if one talks to people in the pharmaceutical industry. But there are aspects of screening that one can think about doing with C. elegans that are different from and complementary to what one can do with more traditional approaches. In fact, I founded a biotechnology company some years ago where one of our approaches was to use C. elegans for drug screening. We had some interesting programs. We did not develop any drug that was commercially successful, but that frankly is true of almost every biotechnology company you've ever heard of. And I think it is still a viable and interesting prospect to use C. elegans and other organisms. People are using yeast, people are using fruit flies, people are using zebrafish in this way. But one has to be careful not to be naive. The pharmaceutical industry has some very sophisticated approaches to drug screening. And so if one is going to do this, one wants to do it with someone experienced nearby to know what the issues are as you do screening, be it low throughput or confirmatory or high throughput. But I think there are certainly potentials, and I actually have another company now that is pursuing exactly this goal, and we have yet to see if it's going to be successful in that sense.
Sorry, the last one is, can you comment on the mechanisms which determine the timing of events in organ development and activation of organ cell death, and the role of epigenetics in this development?
Yes, I could probably talk about that for many hours. So the control of developmental timing is a crucial problem in developmental biology. And in fact, my laboratory identified the first genes and gene pathways in this area. We labeled the genes heterochronic, and that's basically an analogy with homeotic genes. Homeotic genes, when mutated, cause spatial transformations in cell fates. Homeotic genes control spatial cell identities. Heterochronic genes, when mutated, cause temporal transformations in cell fates, i.e. these genes define the developmental timing of specific cells. And what we postulated many years ago was that the heterochronic genes we had identified in C. elegans, when one looked at the nature of their effects on cells, were likely to be cancer genes. Because, for example, they would make cells at a mature time, a late time of development, behave as if they were immature and continue dividing when they should stop dividing. Exactly what cancer cells do. The people in my laboratory who were most responsible for the early studies that we did were two postdoctoral fellows, Victor Ambrose and Gary Rufkin. They went to their own laboratories and they molecularly characterized these developmental timing genes. And what they discovered was that some of these genes encode not proteins, most genes encode proteins, but rather tiny RNAs. RNAs today that are known as microRNAs. And microRNAs are one of the most important, in my view, emerging fields in modern biology. They are key biological regulators, and they also are clearly important in a variety of diseases, most notably in cancers. So the control of developmental timing has now been connected to microRNA genes and cancer. And I think if you watch, you will find this area becoming more and more visible in the very near future as these discoveries continue to be promoted and recognized. Now, epigenetics, you asked about the epigenetic role, is also an area very near and dear to my heart. Our studies of organ development actually led us straight into the biology of epigenetics. And again, I won't go into the details of the science, but the very same set of experiments that led us to characterize the RAS pathway has led us into what is our current focus in organ development, which is genes that are involved in epigenetic regulation. Now, what is epigenetic regulation? If you think about the big problems in biology, the problems are simple to identify. I mean, I would say the first is, what is life? How does life come about? And we've learned the answer to that over the year from bioenergetics and biochemistry and the essence of what makes for a biologically live entity. The unit of life, the cell, has been studied in some detail, and we can go on and enumerate really just a small number of major problems in biology. Without question, one of those problems is heredity. How is it that child resembles parent? How is it that a species resembles others in the same species more than other species? How is it that information is transferred from generation to generation of organisms, but also generation to generation of cells? When a cell divides to make two cells, there's information being transmitted. And many of us thought the answers to these kinds of questions was well known, starting with Gregor Mendel, identifying Mendelian inheritance patterns, and then molecularly characterized as we learned about DNA as the genetic material, the double helix as providing the basis for genetic replication, and gene expression through RNA and protein, translating the information in the DNA to the workhorse proteins that are responsible for many of the biological processes. So genetics seem to be the answer to heredity. What we know today is that genetics is only part of the answer to heredity. The other part of the answer is epigenetics. And by epigenetics, I mean that set of aspects of heredity, of heritable traits, that is defined not by the sequence of the DNA, but rather in some other way. And it turns out that some other way can either be by modifications to the DNA or by modifications to the proteins, the histones that bind DNA. And those modifications can be inherited every bit as much as DNA sequence. And those are epigenetic, those provide the basis of epigenetic inheritance. So my lab today is very involved in the analysis of epigenetic inheritance, and the biotech company I most recently founded is a company founded on translating the emerging science of epigenetics to therapeutics. And because the evidence is now quite good that alterations from normal epigenetic phenomena can be responsible for cancer, and this goes back to the earlier question, yes, there are many ways to trigger cancer, and there also are many different routes to try to intervene. It is looking today like intervention with epigenetic processes offers a brand new route to cancer therapy, and probably to therapy for a variety of other indications, ranging as far as neuropsychiatric disorders. So this company I have founded is now developing drugs. Nope, it's not quite that far, let me reword it, is developing compounds, which we hope will lead to drugs, to intervene with epigenetic processes, and hopefully provide novel treatments for cancer and other disorders. So epigenetics, again, is something, developmental timing and epigenetics are two topics very, very dear to my heart and very, very actively pursued in my laboratory.
Thank you. I think there's a question from the gentleman.
I'm Associate Professor Teo from Sunway University. I'm going to approach this from a slightly different point of view, and what I'm after is an opinion. I was quite interested to hear that when you look at the comparison between what you saw in the worm and what you see in the humans, and your comment was that where humans are concerned, you see families of cells rather than just individual cells. And taking quite a different tack, something that I was curious to throw an idea at and see what your opinion was, comes from the field of suicide. One of the areas that I do a bit of work in is endogenous depression, which is another word for depression that we don't know what the cause is. And very often we're looking for social causes, biological causes, chemical causes, and the list goes on. But nothing seems to work at all, and we see that patients have a will to die. And I'm wondering whether it's such a far-fetched idea that you may have some cells which are involved in programmed death, which may be causing certain parts of the brain to react in a particular way. And why I bring this up is when autopsies are done and scans are done of people who have suicided by endogenous depression, you often see lesions in certain areas. So that's question number one. Is that a far-fetched idea that these families of cells may be clustered in particular areas? The second question I've got comes from the other area which I do a bit of work in, which is psychoimmunology. And in your opinion or in your work, we know that quality of life seems to improve the prognosis of certain kinds of cancers. So translated into what you'd be saying, the quality of life would, I guess, improve the immune system and would stop or reduce the cells from cancer cells from spreading or growing. Therefore, you've got your programmed cell death. Is there at any point in time part of your work which has looked at the role of the environment, at least from a behavioral point of view, in terms of programmed cell death? So that's two questions. First one on suicide, second one on the role of quality of life for the environment.
So in terms of suicide, I think there might be philosophical analogies between the fact that cells can be endogenously programmed to die and the fact that people can be driven in a similar direction. Whether or not there are mechanistic relationships, in other words, are there cases in which a miscontrol of programmed cell death alters, for example, either the brain or maybe the humoral system that, of course, can have great influence over the brain in ways that would drive suicide? I don't know. I haven't seen any data to that effect, but the biology of the effects of programmed cell death in disease is still emerging. So it is not at all impossible, it's not an unreasonable idea that there could be a connection there, but I haven't seen such a connection. I must say that in the studies we've done of C. elegans, we do find analogies with aspects of depression, but the analogies, one always has to be careful not to carry them further than the biology warrants. So that, for example, we have studied very extensively many of the humanly used antidepressants like Prozac, fluoxetine, and many of the others. And these compounds have very marked effect on C. elegans. And if I were to go through my psychological analysis of this worm, I would tell you, and maybe even convince you, that Prozac makes worms happy. Now, whether or not it affects programmed cell death, or one of the hypotheses about fluoxetine, Prozac, and other antidepressants today is that they affect cell proliferation. Whether these effects are seen in C. elegans is not so clear. I would say probably not. But the basic effects of these drugs on the system of serotonin and how serotonin mediates behavior, those analogies are very strong. So I think the bottom line answer is that from the work we've done, we see some similarities in terms of aspects of biology, but I don't know that there's anything in the area of programmed cell death per se that would relate in a way that I could comfortably say it relates to human suicide. In the area of psychoimmunology, again, I think the data that have emerged in this area are quite clear. It's very real that you can have effects on disease progression caused by the way people feel and think. And that makes sense because we are biological creatures and we have all of these complex signaling systems within us, and it's not surprising that those signaling systems in some way can intervene with the course of disease. Do we see effects of the environment on C. elegans? Well, the answer is first order, absolutely yes, but not in this particular context. Okay, so we have not seen environmental effects on programmed cell death. We do see environmental effects on many aspects of worm behavior, and we have in my lab studied these extensively. We analyze how the environment affects behavior. We analyze how experience modulates behavior. But we have not seen any effects on programmed cell death. Does that mean there are none? Probably in this case, but not with certainty.
Thank you.
There's a question on the back, and after that, followed by.
Hi, my name is Aisha. I'm a principal investigator from the medical faculty, and I work on developmental neurobiology. I have a very simple biological question to ask you. The neural tube, as I'm sure many of you know, is the precursor of the brain, the spinal cord. My question is, what animal model do you think is best to study apoptosis in closure of the neural tube? Because there's various evidences in different models. Say, for example, dorsal closure and the drosophila. Paul Martin did that work, and they showed intercalation of cells, but no evidence of apoptosis required. However, in the mouse embryo, for example, we do see a lot of apoptosis all along the neural tube from the cranial region right up to the spinal region by tunnel assay. But whether or not apoptosis is a primary or a secondary effect, I mean, it is still unknown. What would you recommend to be the best model to study apoptosis and dorsal closure and neural tube closure?
Well, my first comment is that that's not a question that I've thought about before, so I would make no dogmatic statement that I have deep knowledge. My preference always is to try to find the organism that is most experimentally tractable, that displays the phenomenon of interest. So if you tell me Drosophila doesn't, but a mouse probably does, I would look carefully at the mouse, and I would consider other organisms like zebrafish, which can be analyzed experimentally in some ways that are easier and faster than with a mouse, and I would see whether or not it's possible to analyze this problem directly with zebrafish. For example, there are many zebrafish developmental mutants. My guess would be that some of those show a defect in closure of the notochord and of the cord in general. And I would look to see if there are such mutants around, rather than trying to isolate them from first principles, and see if there maybe is an experimental system that could be available for use. But I would have to do homework to answer that question.
Thank you.
Prof. Horvitz, so far it's an honor for me to be able to listening to your talk this morning, and also not to hear, because your work has been required papers for us to read when I was doing my PhD in neurobiology and doing from Houston, Ohio. So it's a pleasure to meet you in person. I actually have four questions, but simple ones. Number one, as a developing country in Malaysia, what would you suggest on the proportion of money to be allocated for basic and or fundamental versus applied? You were talking about it slightly this morning. You touched on that slightly. Number two, it's I think has some relationship with a question by Russia just now. In terms of for Malaysia, our main, I think, advantage is being in a tropical country where we have diverse fauna. So there are many organisms that we can actually use for experimental models. And I was just wondering on your thoughts of if we were to use these local species, but sort of replicating things that have been done in a more developed countries. What are your thoughts on us spending time to do that? Some people say you're not going to get anything different, or do you think there will be, whether it's worth spending money and time on that one? Third question is, you mentioned this morning also, but again just now about neurodegenerative diseases as being involved in programmed cell death. I was just wondering how you relate that, because I thought neurodegenerative diseases are diseases of the very mature adult established system, and program is more towards the younger ones. And my final question is very simple, is I read your CV and it looks like you did, of course you're interested in biology way during high school, yet you go into mathematics at econs for your first degree, and you go back to microbiology in fact for your masters, and I thought that the two were just jumps of, we don't do it nowadays. And just wondering of what was the push for those two instances? Thank you very much.
Okay, so 4 questions.
Different.
Some of which are very hard to answer. So how should resources be allocated in Malaysia between basic research and applied research? That is a question of policy. And I do not have enough knowledge of either the science or the needs of the country to begin to try to answer that question in a quantitative way. What I believe very strongly is that if the basic research is too limited, it cannot possibly support the applied research in a sufficient way to allow the applied research to be successful. So there has to be some minimal amount of basic research, and how much that ought to be out of the entire research portfolio is not a question I'm qualified to answer, but my guess is that it is more than people tend to think when they focus on particular applied problems. And that's true not only in Malaysia, but in every other country I've ever been in, including the United States, that you really need the basic as the foundation of the driver. Now, in terms of the ecological diversity, I would say probably not only fauna, but also flora. There is obviously an enormous opportunity here for the identification of novel projects and uses in novel ways of the biological resources. How these resources ought to be used effectively depends upon the problem of interest. So just as an example, if we think about some of the breakthrough advances in modern biology, there are a number that have come from studying organisms that were essentially ignored at the time. My own work in C. elegans is like that. Nobody knew about C. elegans. Nobody thought that C. elegans was likely to be of great interest. The work of Tom Cech using the single-cell tetrahymena led basically to the discovery of catalytic RNAs, a very important fundamental discovery from an organism, again, that most people paid no attention to. So I would look at the species diversity here with 2 different questions in mind. One is, are there species that have some aspect of biology that is so special that you would have an opportunity to solve problems in biology using that species that you couldn't solve in other ways. For example, in neurobiology, you're a developmental neurobiologist. In neurobiology, the electric eel was used because the electric organ has very high quantities of the molecules that are involved in electrical signaling. And the sodium channel, one of the major components of signaling systems in nerve cells, was purified from the electric organ of the electric eel and would have been very difficult to obtain, particularly at the time, in any other way. So I would look at the species and say, what's really different about some of the species that are distinctive, and do these species offer an inroad to an important and unexplored problem in biology? The second question, and a little closer, I think, to what you had in mind as you worded the question, is the comparative question. Oftentimes, when you do comparative biology, you find things that are extremely instructive about biological processes, both how they occur and also how they have evolved. And thinking about evolution, I said before, heredity is one of the main problems in biology. Well, probably the biggest problem in biology is evolution. And the way to understand evolution is by understanding development, genes, and interspecific differences. So if one could choose an appropriate problem and study how an appropriate set of distinctive organisms have solved that problem, one can get potentially very deep insight into both the biological repertoire that's been used and also the evolutionary variation in the processes involved. So those are the two areas I would look for. Unique opportunities to solve particular problems and evolutionarily divergent solutions to similar problems. The third question you raised about neurodegeneration. Okay, so neurodegeneration, indeed, if we think about Alzheimer's disease, we're talking about neurons that have been around for a long time before they start to die. And yet, when I talk about apoptotic death, that's basically a developmental process. So how do we reconcile those? And the answer from what we know of neurodegenerative disease is that it varies and is complicated. So that for Alzheimer's disease, there is still debate in the field the extent to which the cell deaths are apoptotic. Probably it's going to turn out that they have some apoptotic attributes and some attributes that are different, and maybe some cells do one, some do another. But there are other neurodegenerative diseases where the data are very clear that what one is seeing is an apoptotic death. Certain retinal degenerations are unquestionably apoptotic. The scientific data are very compelling that these deaths are apoptotic. They use that same biological programmed cell death that we found in the worm. So I think that we have to separate these neurodegenerative diseases in the same way I said before, we have to be careful to separate cancers. And part of the problem is ignorance. We don't know everything about many of these diseases. In fact, we don't know everything about any of them. But we know enough to know they're not all the same. Alzheimer's is a more complicated disease than Parkinson's. And Parkinson's is in its way more complicated, at least to treat, than in principle than certain retinal degenerations should be. And in terms of apoptosis, again, very compelling data for certain retinal degenerations, suggestive data for a variety of other neurodegenerative diseases. And that retinal degenerations can even be later onset and still be apoptotic in nature. So the timing doesn't look like it's a crucial feature. They can be activated to die later, but that may not always be the case. In terms of my personal history, maybe that's the hardest question of all to answer, but it should be the easiest. In Indeed, in high school I did a project in genetics, and I have ended up professionally as a geneticist. But I did diverge for a while and explored other possibilities. And certainly as an undergraduate, I received 2 degrees, one in theoretical mathematics, one in economics. My minors were computer science and psychology. Okay. I had no exposure to biology until I was in my last year. Why did I change? Probably a number of reasons. One is I was curious to learn something that I knew nothing about. 2 is that at the time, I didn't feel, this was at a time in the late 1960s, I didn't feel that the work I was doing in either mathematics or economics was likely to have the potential impact of what I might be able to do in science, and particularly in biology. I didn't know enough biology to really even think about it critically, but my sense was that biology offered the opportunity to make discoveries that could have an impact, at least on a shorter time scale, in theoretical mathematics. Plus, I wasn't as much a mathematician as some of the people I knew. So I decided to go to graduate school in biology quite literally as an experiment. And maybe that underscored the fact that I'm an experimentalist. And I did it thinking that if it didn't work, I would do something else. And I tried it, and I liked it. I thought, as a senior in the introductory biology course, I went to my professor, a man named Cy Leventhal, and I said to him, Dr. Leventhal, I'm majoring in mathematics and economics and taking your course in introductory biology. I've now read 6 weeks of the textbook, and it seems interesting to me. I'm thinking about applying to graduate school in biology. Am I crazy? Is this too late? And he looked at me and he said, my undergraduate major was physics. My PhD was physics. I'm teaching your biology course. You're starting early. There's always opportunity to do something new.
Thank you. I think in the interest of time, I may have to limit the number of questions. I think there's a lady there who has asked one and another gentleman over there. If you could keep your questions short, we might be able to distribute the time a bit better. But I think we have to let the lady take first. She put up her hand up.
Very good afternoon, Professor, ladies and gentlemen. My name is Tan. I'm from University Science Malaysia, Penang. Now, I'm sure, Professor, you agree with me that program cell death does not just limit to the hypothesis. It also covers autophagic cell death, and some people say program necrosis. Now, there is some hypothesis, the recent hypothesis that actually say or suggest that both apoptosis signaling pathways and autophagic pathways are intertwined, they are related. So if a cell destined to die by, for example, apoptosis, if you stop apoptosis, it will die through the autophagic cell death. So is there such a thing as this so-called molecular switch as suggested by some of the researchers? If there are, what are the ways that we can look at it? Thank you.
I'm not sure I can add much to what you've said. What you've said is totally accurate. And there clearly are other mechanisms that can cause cells to die. And in fact, one of the very active areas in my laboratory is exploring some of the mechanisms that cause cells to die in C. elegans that are independent of this core pathway for programmed cell death that I outlined before. But how these different processes are interrelated, I don't think I know any more about than anybody else. And this is, I mean, what can you say about science besides the fact that the more that you learn, the more you learn that you want to know. So there is a world yet to emerge about the problem of cell death and its regulation, and I think we will of organisms to help inform that world. But what you said is totally right, and I don't have any insight to add beyond that.
Thank you. I think that gentleman with the right arm.
So my name is Dr. Samir. I'm from Faculty of Biology, and I have two questions to the respective Prof.
You may have to limit it to one because there are a lot of others waiting.
My first question is, can the mechanism of thought process be influenced and controlled by external influences? And if some biologists wish to control it themselves, is it achievable? My second question is, can the insight which is obtained from studying mechanism of the thought process be utilized towards achieving self-regeneration?
Okay, I'm sorry, I didn't understand the first question again.
So can the mechanism of the thought process be influenced and controlled by external influences? And if some biologists wish to control it themselves, is it achievable and how?
Okay, so I think the answer to that actually is very similar to the comment made before about cancer. There are many, many pathways that can influence apoptotic death. And these pathways essentially converge ultimately on this core common root. But yes, there are lots of influences, including radiation, chemotherapy, which are used as therapeutics in cancer. They're activating the apoptotic process, programmed cell death. There are other exogenous factors that can do so. There certainly are xenobiotics, small molecules that can influence. So there's no question that there can be a variety of external influences on programmed cell death in any of a variety of organisms, including us.
And my second question is,
maybe we should take the question from the gentleman there.
Oh yeah, hello. I'm from IAS. I'm an A-level student, and I wanted to ask, now that a lot of bacteria that cause disease are getting resistant to antibiotics, what do you think about the prospect of using bacteriophages in the fight against disease? Thank you.
Again, complicated question with a long history. The idea of using bacteriophages to fight bacterial diseases has been around for more than 80 years. And there have been ever more sophisticated approaches to this problem. Some of the recent approaches that are being taken that I've heard about, is this still on? It's running out. Okay. How about like this? Like it? No, it's running back. It's okay. Some of the approaches I've heard about to do this are very clever and sound promising. But historically, this has been a difficult problem. So until I see real clinical data, I'm going to keep open-minded about the promise of this approach. But I think, certainly you look at some of these resistant bacterial forms, and you want to find novel ways to treat them. But so far, I have not yet seen the clinical data that are compelling that the bacteriophage treatments are going to work. But I have seen company business plans that sound very promising, but we'll see.
I will allow one more question. That would be cutting into summing up time, but I'm sure you don't mind. Yes.
Good afternoon, ladies and gentlemen. It's an honor to be here. Thank you, Mr. Professor Horvitz, for this opportunity. I am Mohammed Nasser from Sudan, former physician, currently seeking a degree in economics. So I was actually fooled by the title of the lecture, hoping it would be in regulation of organ. In regulation, I mean the political regulation and the legal aspect. Anyway, I'm going to ask my question to what I believe the lecture would be, or the dialogue would be. And it's in patenting discoveries, patenting especially pathways and genetic material that has been discovered. So what do you say about that? Should it be patented? And should one company or should one organization be allowed to monopolize that discovery for a foreseeable future? And how can we prevent these discoveries from actually, you have been talking about these discoveries being used in therapeutic ways, but being not naive, I think it can also be used in other ways, destructive ways. So what approach should be used to prevent these discoveries from being used for destructive ways? Thank you very much.
So, patents. The goal, is this still on there? It's fading. So let me try.
The goal of patents should be to promote the development of appropriate uses. And sometimes patent laws and patent policies are put in place that don't do that in the most proper of ways. So that a patent can be good if it allows the corporate development of an idea that takes such expense that without protection, that idea would never be brought to a point of being useful. A patent can be bad if it's preventing, essentially, the effective use of knowledge that would then lead to discoveries and the development of applications. So that, for example, one of the things I was involved with some years ago was developing for the Genome Institute of the National Institutes of Health in the United States a white paper about the patent policies concerning the patenting of genes. Okay, because of rules that have been put in place prior to the revolution in molecular biology, it turned out that it was possible to patent genes. Okay, and that, I think many people would agree, was not promoting the development of drugs and other applications of the genes, but rather curtailing it. And the genes themselves, you know, the fact that they were considered to be inventions, because a patent is for an invention rather than a discovery or even just a description, said that the way the laws were drawn really should have been modified. So patents are good when they are applied in the most appropriate way to allow what is needed for, basically for a society. And so what's important is that, one, patent laws be drawn up by people who are understanding, who are savvy about the issues, and also that they're not done in a way that becomes very difficult to modify as knowledge changes. So I've seen very closely patents that are good and patents that are bad. There are a lot of discussions today about whether the pharmaceutical industry is going to be able to continue to innovate drugs if there isn't sufficient patent protection to allow them to make money after they develop drugs. But there are arguments on both sides. They are complicated, and I think that's more than we can go into now, but I think you can see where the issues come from. Your second question has to do essentially with the use of scientific knowledge. And knowledge in biomedicine is no different from other scientific knowledge, or perhaps more generally from knowledge itself. Knowledge can be used for good or for evil. And it is up to individuals and societies to figure out how to best make knowledge used in positive ways. I don't believe that we want to stop the advance of knowledge. Knowledge offers wonderful things for humanity, such as the development of new drugs. But yes, biological warfare, use of agents in ways that can be very harmful and in fact ultimately destructive of societies and even the world. This is very bad. So we need to have leaders and we need to have societies who are very cognizant of these issues and who can help guide us as we make our advances to use them appropriately. And we have to have scientists who also are responsible and take responsibility for their discoveries and decide which things they want to pursue and which things they don't. But sometimes one doesn't know ahead of time about how a particular body of knowledge can be misused. So we have to have safeguards that go well beyond the individual scientist.
Thank you. I'm sure all of you agree with me that Professor Horvitz has done an excellent job in this dialogue. I'm supposed to have 15 minutes of summary time, but I have sacrificed all of it. So I will have to sum up into 3 seconds. But basically, I think all of you will recall that we really has covered the ground that's quite amazing. Ranging from science policy, the role of scientists in science policy, the importance of nurturing young people, influencing them, inciting them through projects, through doing things and discovering rather than just teaching them facts and information. We have also delved into actual basic science, I would believe, by discussing things like timing genes and microRNA genes, biology of epigenetics, even ranging into the possibility of psychoimmunology, comparative biology, neurotube defects, neurodegenerative diseases, and all that. And finally come back to a very basic and very important aspect of science, and that is the responsibility of scientists for what they discover and how they use science. And finally, I think that would be a very important take-home message, besides just discovering the world around us, that we should be responsible for the world around us. I was going to ask a question, but I think I have to save it till dinner time. But I could give Professor Horvitz a little bit of a clue as to what I might trouble him about this evening, and that is whether more scientists and Nobel laureates should go into politics. I would like you to join me in thanking Professor Horvitz.
Thank you, Professor Horvitz. Thank you, Professor Horvitz, for an interesting and insightful dialogue, and Professor Louis for moderating the session. Ladies and gentlemen, hold on. We would now like to call upon Yang Berbahagia Dato' Engineer Dr. Haji Ahmad Zaidi Laidin to present a memento of appreciation to Professor Horvitz on behalf of the Academy of Sciences Malaysia. With that, ladies and gentlemen, we have come to the end of this session and hope that you have benefited much from the discussion and sharing of ideas. Everyone is cordially invited for refreshments on the ground floor, seminar room A and B. We look forward to your participation in other forthcoming religious events. Thank you and have a nice day.